Do you see it okay?
Yeah.
Okay.
For my screen, it's all black. I don't know if that's,
I
don't know why.
Let me try it again.
Andrew's here.
How's it now, Allison?
I can see the slides now.
Perfect.
James is here.
So we'll just wait like 2 more minutes. All right, let's get started. It is my distinct pleasure to announce the PhD defense for Michael Conley. Michael is my first graduate student. So for me, this is particularly special because Mike has had the, I guess, privilege of being the, of going through this journey of learning how to be a advisor and learning how to you know, navigate this system. So it's a lot of complicated feelings with announcing his defense. So I want to first start by bringing you all on a little journey of little Mikey C here. He has always had a love of the ocean and is known for his ability to build and maintain large aquarium systems and has always had a love of that. And he did his undergrad at University of Miami. So he's been at Rasmus for a while. And this is a picture of him on his beginning his journey to University of Miami. And so then once he was at Miami, you know, it was really exciting because he could start to pursue his love of marine biology and aquarium sciences and really get into diving and actually start to see in their natural environment the animals that he had been maintaining at home for so long. And I would say that his journey started out really innocent. So he was in the band of the Hour. He was a member in that. He was the president of the aquarium club. So he was very, you know, on the straight and narrow, very academic and really, you know, focused on learning about marine science and playing in the band. But that like quickly changed. And I think he really embraced the Miami lifestyle. And yeah, Mike, I had to include something like this. So and so, you know, there's what's really interesting to me about Mike is that there are a lot of like hidden dimensions and things that I'm still learning about him, even though I've now known him for over 5 years. But then he started graduate school. So he calmed down a little bit and kind of regrouped. He helped with, you know, he led the building of the aquarium system in my lab. And he has been extremely successful and is an award-winning speaker. He's won many awards for his seminar presentations, as well as he was People's Choice Award. He won that for the 3-minute thesis. And what is so delightful to me about Mike, especially in this context of being a speaker, is that he's so humble about this. So, you know, he's won all these awards, and yet every presentation, he's still nervous as though it's his first one. You know, I appreciate that a lot about him, but he is an award-winning speaker. But he's still having like a really good time in graduate school. And I think a lot of that has to do with his relationship with Anne and the support and you know, love that she has been able to give him. And it's been really, really exciting to see him grow, not only as a scientist, but also as a person. It's like really, really wonderful to see. And, you know, like I was saying before, he's just had a lot of accomplishments. I mean, he is a tough act to follow. So he has had 5 published papers and 3 are in review and revision, and there's probably going to be a couple more that will come out of his graduate work and all the collaborations that he's had. He has been awarded 7 research grants and scholarships, including from the Marine Aquarium Societies of North America, through the University of Miami, the Global Invertebrate Genomics Alliance, as well as NSF. And the funny story about that award is that he was awarded that award, I believe in your second year, Mike, right? Or the end of your first year? Mm-hmm. Yeah, and so he was just starting, and we said, okay, apply for this. You should go to Taiwan and run experiments. So Mike, he got the award and went there alone and ran all these experiments. And a lot of these experiments are now the work that he'll be presenting and have built his thesis. And so to me, that shows just how independent and smart Mike is. He was willing to take that chance to go to a totally new country, working on a totally new project and system, and be really successful with it. And so today he's going to be talking about the interactions between the Pocillopora coral innate immune system and coral-associated bacteria communities. And this was the work that he started in Taiwan and then brought it to Rasmus and has expanded on this. And it's been really, really gratifying and wonderful to see Mike grow into this amazing scientist with this and becoming this expert on coral immune systems. And lastly, I'm extremely proud that Mike will be starting a postdoc at the Smithsonian based in between DC and Panama as a Smithsonian Biodiversity Genomics postdoctoral fellow, where he'll be continuing the work and expanding upon the Pocillapara damicornis work that he started as a graduate student. And then lastly, I just want to say on a personal note, Mike, I just really have appreciated being able to be your mentor, to work with you and to get to know you. I'm so proud of you, and I'm getting a little choked up. I'm just really, really proud of you. So yeah, I can't wait to see what the future holds. And thank you so much for being a wonderful grad student. And with that, I pass it over to you to start your defense.
Mm-hmm. Well, thank you, Nikki. That was really, really touching, and I really appreciate that introduction. Can everyone see my screen all right? Okay. Well, good morning, everyone. Thank you all for being here. Today, I'm going to be talking about some of the research I've been working on throughout my PhD, investigating interactions between the Pocillopora coral innate immune system and their associated bacteria communities, and trying to understand how these coral bacteria interactions influence coral health and resilience. But first, I want to tackle a question I get a lot from my family and friends, and that is, many of whom are on the call, and that is, what are corals and why am I so interested in them? Well, corals are animals in the phylum Cnidaria, meaning that they're closely related to sea anemones and jellies. But to me, they're just incredibly beautiful animals, taking many different forms with magnificent colors, shapes, and sizes. But what really drew me in deeper into wanting to study corals in graduate school was when I realized their immense ecological and economic importance for both marine ecosystems and human society, and how the complexity of corals' microbial symbiotic relationships affects coral health and underpins their success throughout the world's tropical oceans. So then, why do corals matter? Put simply, this is because corals, like this little Pocillopora colony on the reef near Wanlitung village in southern Taiwan, build up the coral reefs that support the world's most biodiverse marine ecosystems, sustain productive food fisheries, protect shorelines from waves and storms, and drive tourism economies for coastal communities worldwide. And I think you can see all the reasons why coral reefs are so important in this aerial photo I took of Wanlitung village during my field research in southern Taiwan. You can see the modern coral reef growing out along the coast and the small village that has sprung up next to it, with a few seafood restaurants, snorkeling shops, and hotels. However, the cliff in the background is also a fossil coral reef, which was uplifted over 3,000 years ago. I think this image captures just how important corals and the reefs they build are, both in our present moment and throughout the Earth's history. Okay. However, corals are in crisis. Global climate change caused by mankind's greenhouse gas emissions has resulted in an ocean that is becoming hotter, more acidic, and less oxygenated. Local stressors such as nutrient runoff and pollution can also diminish coral health. Corals respond to these many stressors by bleaching, which is essentially a breakdown in corals' microbial symbioses, which eventually can kill corals and destroy entire reef ecosystems. When corals die, the physical structure of the coral reef starts to degrade, leading to the loss of habitat for reef fish and invertebrates, and threatens the loss of ecosystem services for the human communities that depend on them. This photo is of the reefs in Taiwan during the severe coral bleaching that occurred there this past summer. So then, why are host-microbe interactions important for our understanding of both coral health and biology in general? It's important to recognize that microbial interactions are everywhere and are fundamental processes in biology. Sometimes I like to say, it's a microbial world and we're just living in it. In our case, we're often interested in this endosymbiosis between corals and microalgae in the family Symbiodiniaceae. But it's also well known that animal host bacteria interactions influence invertebrate traits, ecology, and evolution, such as in the model cnidarian hydra, which has a simple immune system that it uses to regulate its gut bacteria, the diverse insect mycetocyte symbioses, in which specialized bacteria with metabolic complementarity to their host are maintained in specialized cell types, and the well-studied endosymbiosis between the Hawaiian bobtail squid and its light-producing bacteria, Vibrio fischeri. Next, what is the role of the innate immune system in regulating these diverse host bacteria interactions? Generally, the classic view of the immune system suggests that its role is to eliminate pathogens and maintain homeostasis with commensal microbes. These days, we've been hearing a lot about how the human immune system helps protect us against diseases such as COVID-19. And in invertebrates, the immune system is also responsible for protecting the host against pathogens. However, it's important to recognize that invertebrates lack the ability to produce antibodies and generate highly specific immune memory, as in the vertebrate adaptive immune system. And so they must rely on the activity of the innate immune system to defend against microbial invaders. Invertebrate immune systems generally achieve this by the recognition of microbe-associated molecular patterns, or MAMPs, by different classes of pattern recognition receptors, PRRs. The binding of these MAMPs to PRRs activates intracellular signaling pathways that cause inflammation and enhance immune gene expression. The mounted immune response then involves effector mechanisms that perform microbial killing and clearance, such as phagocytosis by specialized immune cells, melanin deposition, and the production of antimicrobial peptides in reactive oxygen species. Together, these processes are known as the recognition, signaling, and effector phases of innate immunity. To comprehensively study coral innate immunity and host microbe interactions, we turn to corals in the genus Pocillopora as a model system for two key reasons. First, Pocillopora corals have a broad geographic distribution and a natural range in the Indian and Pacific Oceans from East Africa to the west coast of Central America. These are some photos of different Pocillopora colonies at field sites I visited throughout my PhD, spanning the Pacific from Asia to Central America. Second, Pocillopora corals tolerate captivity better than other species and are easy to culture in aquariums. We can also create small, replicate fragments for use in complex manipulative experiments. In addition, Pocillopora corals associate with a large diversity of microbes, which are altogether referred to as the coral holobiont. The well-studied endosymbiosis between corals and Symbiodiniaceae algae involves the transfer of photosynthetic products from the algae to the coral host in exchange for shelter and other nutrients. Pocillopora corals also associate with bacteria that we are now beginning to understand benefit the health of the coral holobiont through various mechanisms, including additional carbon fixation, nitrogen cycling and amino acid synthesis, sulfur cycling and antioxidant functions, B vitamin provisioning, and the production of protective antimicrobial compounds. In Pocillopora corals specifically, gamma proteobacteria of the genus Endosymbiotic have been shown to dominate the bacteria community in many studies and have been found as cell-associated multicellular aggregates, or commas, within coral tissues. And even more surprising, very recent work has shown that Symbiodiniaceae are also intimately associated with diverse bacteria that live next to or even inside individual algal cells, even while the algae are inside coral host tissues. All these partners, as well as other viruses, fungi, and micro-eukaryotes that are present, collectively form a holobiont community that consists of numerous multiple partner nested symbiotic interactions, with diverse interactions all occurring within coral host tissues. These interactions occur across a spatially heterogeneous landscape that includes the surrounding seawater, the coral mucus layer, and two tissue layers, the epidermis and gastrodermis, where Symbiodiniaceae cells are maintained within coral gastrodermal cells. To help mediate these complex microbial interactions, corals must rely on the activity of their innate immune system, which has been shown to possess several classes of evolutionarily conserved pattern recognition receptors, signaling pathways, and effector mechanisms. Likewise, shifts in microbial community composition caused by environmental factors can alter coral immune status, such as when high temperatures cause corals to expel Symbiodiniaceae from their tissues, leading to coral bleaching and increased risk of infectious disease. My research involves the use of different treatments with common MAMPs as immune stimulants, as well as broad-spectrum antibiotics and short-term heat stress as experimental tools to activate coral immune responses and disrupt corals' interactions with their bacteria communities, and then measure the downstream effects on coral gene expression and bacteria community shifts. Which brings me to my three key research questions I'll be discussing today, where I'll be presenting evidence and data from three of my dissertation chapters. First, how do Pocillopora corals interact with their associated bacteria communities? Second, how do Pocillopora associated bacteria communities influence holobiont heat tolerance? And third, how does the disruption of Pocillopora associated bacteria communities affect coral holobiont physiology? To address these first two questions, I will describe the results of two experiments I completed during a National Science Foundation East Asia and Pacific Summer Institute Fellowship in Taiwan during the summer of 2017. Why did I choose Taiwan? It is located at a similar latitude to Florida and is near the Kuroshio Current, a warm water western boundary current similar to the Gulf Stream, and has fringing coral reefs with high coral diversity and abundant Pocillopora corals, which are unfortunately not present in the Caribbean. I learned a lot about Taiwanese culture during my orientation week in Taipei, and then I traveled by train to Hengchuan to work at the National Museum of Marine Biology and Aquarium, or NMMBA, which is both a gigantic public aquarium and a top-tier marine lab all in one, and was a great place to do this research. For my experiments, I collected Pocillopora corals from 2 sites on the Hengchuan Peninsula, Howan and Wanlitung, and brought them back to outdoor flow-through seawater aquariums at the NMMBA. Here are some photos that show how we located the colonies, how the collection process worked, and the flow-through acclimation aquariums, and a close-up of one of the corals. I collected 2 Pocillopora colonies at each site, and after sequencing, these would turn out to be 4 different genotypes from 3 different cryptic species. Howan 1 was Pocillopora damicornis type 8, and Howan 2 was Pocillopora damicornis type 4, or type alpha. At Wanlitung, both genotypes were Pocillopora acuta, or type 5. Our current understanding of the Pocillopora species phylogeny suggests that these species are closely related and diverged only recently, but this distinction is important for how we interpret the gene expression and bacteria community data. The different shapes, circle, triangle, rectangle, and diamond, represent each genotype on the following plots. For my first experiment, I subjected replicate fragments of these 4 coral genotypes to a control treatment and a 4-hour experimental exposure to a common bacterial MAMP, lipopolysaccharide, or LPS, in order to stimulate the coral immune response. LPS is typically found in the outer membrane of gram-negative bacteria and is a conserved molecular signal that has been used to stimulate immune responses in many previous studies on humans and non-model invertebrates. I completed this design using 3 fragments of each genotype in each treatment for a total of 24 fragments, and then preserved samples at the end for coral RNA-seq analyses. I found that LPS exposure activated Pocillopora immunity, but that overall transcriptome patterns were species and genotype specific, as you can see in this principal components analysis plot. However, after accounting for genotypic variability, there were 395 differentially expressed genes between the control and LPS treatment, and upregulated genes, highlighted in red on these next 2 plots, included toll-like receptors and scavenger receptors, immune-related transcription factors and signal transduction genes, and pore-forming toxins such as perforin 2. Downregulated genes in blue were involved in ammonium transport, antioxidant functions, and neurotransmitter metabolic processes. The heat map of differential gene expression shows that while overall expression patterns were indeed genotype specific, Pocillopora corals from different cryptic species still share a conserved immune response to LPS. Notably, 2 coral genes that were upregulated in response to LPS are homologous to known pore-forming proteins with critical roles in human and invertebrate immunity. The first was similar to perforin 2, which is a protein that is essential for bacterial killing in human, mouse, and zebrafish macrophages, and was discovered here at the University of Miami's Miller School of Medicine by Dr. Eckhard Podek's group. Perforin 2 possesses a membrane attack complex pore-forming domain that inserts into bacterial membranes to cause lysis and cell death. Interestingly, all the Pocillopora genotypes also upregulated another MACPF domain-containing protein that was similar to a toxin characterized from the Okinawan sea anemone Phylodiscus simoni. Studies from the early 2000s showed that this protein could activate the complement pathway and possess lytic activity, suggesting its role as an important cnidarian immune effector mechanism. Lastly, searches through other cnidarian and coral genomes revealed that this family of toxins has undergone multiple gene duplication events throughout coral evolution, and that Pocillopora alone has 19 of these important effector proteins. Altogether, analysis of the Pocillopora genome and transcriptome results from LPS treatment indicate that Pocillopora corals have a complex and diversified innate immune system, capable of executing the recognition, signaling, and effector phases of innate immunity. Furthermore, the high level of diversification among coral PRRs, signaling pathway components, and antimicrobial proteins suggests that corals may be capable of more fine-scale discrimination of different microbial signals and subsequent modulation of immune responses than previously considered. So once we understood that Pocillopora corals possess a diverse innate immune system that responds to bacterial MAMPs, such as LPS, we wanted to try to understand how disruptions in coral bacteria community composition affect the activation of these immune factors, as well as the overall holobiont response to heat stress. For my next experiment, I subjected replicate fragments of the same 4 Pocillopora genotypes to different experimental treatments, including the same control treatment in seawater at the summertime average temperature of 28 degrees Celsius, and acute 48-hour heat stress at 32 degrees Celsius, which is 4 degrees warmer than the control. And I also exposed fragments to 100 micrograms per milliliter of the broad-spectrum antibiotics ampicillin and streptomycin for 72 hours. These antibiotics were chosen based on their different modes of antibacterial action, and because these concentrations did not cause visible stress to Pocillopora fragments during preliminary experiments. Finally, I conducted combined treatments with antibiotics exposure and heat stress, so I could examine the additive effects of these different stressors on coral and Symbiodiniaceae gene expression and bacteria community responses. I completed this design using 3 fragments of each genotype in each treatment for a total of 48 fragments, and then preserved samples for the dual RNA-seq and bacteria community analyses. Previous research had shown that the coral heat stress response is affected by the composition of the bacteria community, and that antibiotics treatment of Pocillopora corals from Australia diminished fragment heat tolerance and caused death in combined treatments, when no corals died in the separate antibiotics or heat stress treatment. In this study, the authors hypothesized that antibiotic treatment causes a shift in the coral bacteria community away from beneficial bacteria that results in disease and death, or what can sometimes be referred to as dysbiosis, during a subsequent heat stress. My results support these observations and provide additional information about how coral and Symbiodiniaceae gene expression is linked to these shifts in bacteria community composition. First, I wanted to show some representative pictures of the fragments taken at the time of sampling. You can see some slight morphological variation in the control fragments of each genotype, and that there were no major differences in fragment condition in either the separate heat stress or antibiotics treatment fragments within each genotype. However, fragments in the combined antibiotics heat treatment appeared visibly stressed at the end of the experiment, and some had signs of local tissue loss, as seen in this genotype and Wanli-Tung-2, this fragment from Wanli-Tung-2. Next, RNA-seq analysis of Pocillopora and Symbiodiniaceae gene expression revealed that both partners react more strongly to the antibiotics heat treatment than the separate antibiotics or heat stress treatments. After determining that each of the coral genotypes was dominated by Symbiodiniaceae in the genus Cladocopum, I aligned the RNA-seq reads against the Pocillopora and Cladocopum reference genomes and proceeded with gene expression analyses. As you can see, approximately 69% and 16% of the total reads produced from the experiment aligned to either the Pocillopora or Cladocopum genomes, which is useful for more informative downstream analyses of holobiont responses to these stressors. The principal coordinates analysis plots show that overall transcriptome patterns are still host genotype specific in both Pocillopora corals and their Cladocopum algal symbionts. However, there are clear effects of experimental treatment, including a strong additive effect of the combined antibiotics heat treatment on both Pocillopora and Cladocopum gene expression. The colors in these plots are blue for the control treatment, orange for the heat treatment, cyan for the antibiotics treatment, and yellow for the combined treatment. And the treatment centroids are plotted as black circles outlined in each treatment color with lines connected to each sample within the treatment. Interestingly, antibiotics treatment has a stronger effect on gene expression in Cladocopum than in Pocillopora, suggesting that changes in the coral bacteria community have more pronounced effects on Symbiodiniaceae within coral tissues than on the coral host itself. This pattern is also observed in the number of differentially expressed genes, or DEGs, detected among experimental treatments when contrasted against the control. When we examine these Venn diagrams, we can see that thousands more differentially expressed genes are detected in only the antibiotics heat treatment rather than in the individual antibiotics or heat stress treatments for both Pocillopora and Cladocopum. We can also see many more differentially expressed genes in the Cladocopum response to antibiotics treatment compared to only a few dozen differentially expressed genes in the Cladocopium response to heat stress. When we examine which biological functions are enriched among the sets of upregulated and downregulated genes, we can see that Pocillapara and Cladocopum have different functional responses to the various experimental treatments. These are bar plots representing enriched eukaryotic orthologous group, or COG categories, for both Pocillapara and Cladocopum upregulated and downregulated genes in each of the treatments versus the control. COG categories are meant to represent the major functional classes of genes present in model eukaryote genomes. COG categories are shown on the vertical axis, and the horizontal axis depicts the enrichment score for each COG category within each set of upregulated or downregulated DEGs. An enrichment score above the dashed line means that the biological processes in a given COG category are significantly overrepresented in the list of upregulated or downregulated genes for a given treatment. For Pocillapara, we can see that downregulated genes in the heat stress treatment were enriched for the COG categories translation, ribosomal structure and biogenesis, and inorganic ion transport and metabolism, which is consistent with other studies of the coral heat stress response. Pocillapara downregulated genes in the antibiotics treatment were also enriched for inorganic ion transport and metabolism, while upregulated genes were enriched for intracellular trafficking, secretion, and vesicular transport, and post-translational modification. Pocillapara downregulated genes in the antibiotics heat treatment were enriched for the COG categories amino acid transport and metabolism, cell membrane biogenesis, extracellular structures, inorganic ion transport and metabolism, DNA replication, recombination, and repair, and signal transduction mechanisms, while upregulated genes in the antibiotics heat treatment were enriched for energy production and conversion, intracellular trafficking, translation, and post-translational modification. Altogether, this suggests that the Pocillapara coral host reacts strongly to antibiotics heat treatment through increased energy production and protein modifications at the expense of normal cellular metabolism and housekeeping functions. For Cladocopum, downregulated genes in the heat stress treatment were also enriched for the COG category translation, ribosomal structure, and biogenesis, while downregulated genes in the antibiotics treatment were also enriched for energy production and conversion and inorganic ion transport and metabolism. Inorganic ion transport and metabolism genes were also significantly downregulated in the antibiotics heat treatment, and the downregulation of these inorganic ion transport and metabolism genes is what is driving the observed Cladocopium response to antibiotics, indicating that disruptions in the coral bacteria community may directly affect Symbiodiniaceae specific processes. Lastly, for this chapter, analysis of the Pocillopora bacteria community composition for each genotype in each of the treatments revealed that antibiotics treatments do alter the coral bacteria community and have the effect of reducing overall bacteria diversity. This PCA shows a clear separation of antibiotics-treated samples along the first principal component axis, and this box plot of bacteria richness shows that samples in both antibiotics treatments have lower bacteria alpha diversity on average than the control or heat stress treatment samples. This variation between the antibiotic-treated and untreated samples was also largely driven by a shift away from communities dominated by bacteria in the family Rhodobacteriaceae and towards bacteria in the family Alteromonidaceae. You can see in this relative abundance bar plot that putatively beneficial bacteria in the family Rhodobacteriaceae and genus Endosymbiosis are decreased during antibiotics treatment, and potentially opportunistic microbes in the genera Alteromonas and Vibrio proportionally increase in relative abundance, including some strains with 16S rRNA gene similarity to known ampicillin-resistant bacteria. To summarize what we are observing in this experiment, the current model of coral bleaching involves a healthy coral subjected to a prolonged heat stress that compromises the symbiosis between the coral and Symbiodiniaceae, leading to coral bleaching and the expulsion of the algae. After this, the immunocompromised coral is often more susceptible to secondary infection by invading bacterial pathogens. Another possibility is that environmental stress, whether abiotic factors such as temperature or pollutants, or biotic factors such as bacterial or viral pathogens, can cause shifts in the endemic coral bacteria community that can then break out into active virulent disease or dysbiosis. In this experiment, we show that antibiotics treatments can be used to cause shifts in the endemic coral bacteria community relative to fragments in the control treatment that maintained their normal bacteria, and that the coral and Symbiodiniaceae transcriptomic response to a subsequent heat stress differed when the bacteria community was shifted. This suggests that the endemic coral bacteria community has functional roles in mitigating the severity of the coral host and Symbiodiniaceae response to heat stress, and may contribute significantly to coral holobiont resilience to other environmental stressors. Our results build upon the Gilbert et al. Study by showing that the opportunistic bacteria involved are likely in the genera Altromonas and Vibrio, and that both coral and Symbiodiniaceae gene expression are more strongly affected by the combined antibiotics-heat treatments. Finally, after analyzing the results of both the experiments I completed in Taiwan, I wanted to do one more experiment in Miami to address some more fundamental questions about how Pocillopora-associated bacteria affect coral holobiont physiology under normal conditions, all while analyzing a more diverse cohort of Pocillopora coral genotypes. For this final experiment, I wanted to try and manipulate the bacteria communities of Pocillopora corals that were available at Rasmus thanks to the work of Drs. Peter Glynn, Anna Palacio, and Andrew Baker in importing them from Panama, and also Phil Gillette, who has been culturing these corals in MTLSS aquariums since they've arrived. I selected 14 genotypes, 6 from Uva Island in the Gulf of Chiriquí, 6 from Saboga Island, and 1 from Uraba Island in the Gulf of Panama. These sites exist along a gradient of wind-driven seasonal cold water upwelling, with more upwelling and greater temperature variability occurring in the spring to summer months in the Gulf of Panama. I also chose genotypes with the goal of maximizing the diversity of Pocillopora host lineages and dominant Symbiodiniaceae types from each of these environments, as these traits had already been determined for these genotypes by Dr. Palacio. As you can see from these microscope photos of just 6 of the 14 genotypes, each genotype has distinct morphological features in terms of polyp and skeletal morphology, as well as Symbiodiniaceae density and positions within host tissues. I designed an experiment to determine the baseline bacteria community composition and diversity of each of these Pocillopora genotypes, and also to understand how suppression of this endemic coral bacteria community with antibiotics treatment affects overall holobiont physiology. For the experiment, I subjected samples to a baseline, control, and antibiotics treatment. I used the same 2 antibiotics as in Taiwan, ampicillin and streptomycin, and also included 10 micrograms per milliliter of ciprofloxacin in my treatment because of its potent activity in blocking bacterial DNA replication. From individual parent colonies of each of the 14 Pocillopora genotypes, I created Pocillopora coral microfragments ranging in mass from 80 to 300 milligrams and allowed them to recover for 3 weeks in a flow-through aquarium. This was so that I could maintain these fragments under exposure to either filter sterile seawater or antibiotics-treated seawater in 24-well plates within a temperature-controlled incubator kept at 26 degrees Celsius. Then, for each day of a 5-day experiment with daily media changes, I measured each fragment's dark-adapted photochemical efficiency and oxygen consumption rates, and took fluorescence microscope photos to track fragment condition during the experimental treatments. Using coral microfragments in this experimental design allows me to use the IPAM fluorometer and microplate respirometer to measure up to 24 fragments at once, and allows the Pocillopora parent colonies to heal for use in future experiments. I preserved samples after 5 days for bacteria community and gene expression analyses. However, today I'll be focusing on only the physiology and bacteria community results. So here's a photo of the lab benchtop with the experimental instruments all set up. However, the measurements for each day of the experiment were conducted in the dark. So first, the IPAM results showed that the dominant Symbiodiniaceae type, but not antibiotics treatment, significantly affected Symbiodiniaceae photochemical efficiency as measured by the variable fluorescence over the maximal fluorescence yield. There was a trend towards lower FV over FM in antibiotics treatment fragments hosting each symbiont type. However, this effect was not significant in the statistical model I used to test for these interactions. In contrast, analysis of normalized fragment oxygen consumption rates determined that fragments in the antibiotics treatment had significantly lower oxygen consumption rates than those in the control treatment, a trend that held for genotypes hosting each dominant Symbiodiniaceae type. This indicates that antibiotics treatments impact net holobiont oxygen consumption, either by directly depleting bacteria or by indirectly interfering with coral bacteria and Symbiodiniaceae bacteria interactions in such a way as to reduce the total oxygen consumption by all members of the holobiont. Finally, I want to turn to the bacteria community 16S sequencing results, where we can see that antibiotics treatment has a dramatic effect on Pocillopora bacteria community composition. This PCA plot of Pocillopora bacteria communities in each of the treatments reveals that samples in the antibiotics treatment cluster separately from baseline and control samples and have a narrower distribution along the first two principal component axes. Baseline samples are in light blue, control samples are in royal blue, and antibiotics treatment samples are in light green. And so ellipses represent this 95% confidence interval and show the clear separation of antibiotic samples, whereas baseline and control samples overlap in their distribution. Next, these box plots of bacteria community alpha diversity richness and beta diversity distance to centroids show that Pocillopora associated bacteria community diversity and dissimilarity is significantly reduced in antibiotics treatment, meaning that these communities are less diverse and more similar to one another than the untreated corals. So this is similar to the results from my Taiwan study, and we see that antibiotics treatment ultimately can cause a greater than 50% reduction in overall bacteria diversity, dropping the total species richness from around 120 different bacteria on average to less than 50. When we zoom out to examine which bacterial taxa are reduced by antibiotics treatments in fragments from the Gulf of Chiriquí and Gulf of Panama, we see that baseline and control fragments are enriched in the families Nitroncalaceae, Rhodobacteraceae, Magnetospiraceae, Selvibrianaceae, and Alteramonidaceae, and that each of these families is almost completely eliminated by the antibiotics treatment. In contrast, the less diverse community of fragments in the antibiotics treatment is dominated by bacteria in the genera Endozoicomonas, Blastopyrrolula, and Vibrio, with other representatives from the family Amoebophalaceae and Physosphoraceae. This is interesting because each of these taxa has been implicated in inhabiting more internalized niches within the coral holobiont. Endozoicomonas have been described as cell-associated multicellular aggregates in Pocillopora coral tissues, and the strong signal of Endozoicomonas enrichment here may be because these bacteria are more shielded from the effects of external antibiotics treatment because of their internal location. Similarly, bacteria in the genus Blastopyrrolula and Physosphoraceae have recently been documented in close association with Cladocopum algae, and may resist antibiotics elimination because of their location within the symbiosome compartment of coral gastrodermal cells. Altogether, this experiment shows that we can use antibiotics treatment to substantially decrease coral-associated bacteria community diversity and suppress overall holobiont oxygen consumption without significantly impairing Symbiodiniaceae photochemical efficiency in the short term. This work characterizes the baseline bacteria diversity of Pocillopora coral genotypes in culture at Rasmus and opens the door to more sophisticated experiments that define the functional roles of different bacteria within the Pocillopora holobiont. Now that we've reckoned with the full diversity of bacteria present within the Pocillopora holobiont and used genomic and transcriptomic evidence to assess how the coral innate immune system and Symbiodiniaceae communities respond to different environmental stressors, we can start to piece together a more integrated view of the Pocillopora coral holobiont and suggest avenues for future research to push our understanding of these intricate relationships even further. This work has shown that Pocillopora corals are a tractable model system for short-term microbial manipulation, and future work should aim to characterize the spatial distribution of different microbes within coral tissues. For example, identifying which bacteria form cell-associated multicellular aggregates or are associated with different Symbiodiniaceae types in this same cohort of Pocillopora coral genotypes. Next, metagenomic sequencing would be able to reveal the functional gene content of each of these microbial symbionts, which is essential to better understand their role in diverse holobiont interactions. We also need to better characterize the coral immune factors that manage these microbial partnerships and explore the unannotated coral-specific genes that may have evolved to perform highly specific functions related to coral host microbe interactions. Finally, I think the results of this dissertation place Pocillopora corals in an ideal position to study the processes underlying host immunity microbiome interactions and holobiont coevolution in marine invertebrates. First, we can now work to experimentally resolve the mechanisms of coral microbe interactions and identify specific beneficial functions of coral bacteria and other associated microbes. Next, through careful treatments with both antibiotics and inoculations with live bacteria isolates, we can assess the potential for microbiome manipulation to affect coral traits to enhance resilience and boost adaptive potential, which may be able to proceed on much faster timescales than coral host genetic adaptation. And finally, we can turn back to nature and re-examine the full diversity of Pocillopora corals as not only cnidarian animals, but complex holobionts associated with diverse Symbiodiniaceae, bacteria, and other microbes with integral roles in coral health. The photo on the top right was taken during a recent coral bleaching event in Moorea, French Polynesia, where recent work has identified that different cryptic species of Pocillopora coral had differential responses in bleaching to the same thermal stress event. So by examining the natural diversity of coral holobiont communities with different stress tolerances, we can better understand how coral-associated microbes contribute to coral resilience, adaptation, and diversification along evolutionary timescales. I want to close by reminding you all once again why this research matters in the present moment. Corals are perilously close to the brink, and we stand to risk losing coral reefs as we know them and the critical ecosystem services they provide by the end of this century. However, it's important to remember that corals are resilient and are descendants of an ancient lineage that survives previous mass extinctions and vastly different ocean conditions throughout geologic time in order to construct the enormous coral reefs of the modern day. To ensure corals can survive into the future, we must wholeheartedly combat climate change by reducing our collective greenhouse gas emissions and zealously protect and conserve the few intact, healthy reefs that exist around the world. Next, we need to improve and expand existing reef restoration and intervention strategies, and research into basic coral biology and microbial symbioses will help make great strides towards this goal. And finally, we should aim to support the local communities that depend on coral reefs for food and economic security every day, and work to better educate the public about the importance of being good stewards of our marine environment. So with that, I would really like to thank my PhD advisor, Dr. Nikki Traylor-Knowles, for all her incredible support during these past 5 years. I wouldn't have wanted to work with anyone else on these projects, and I'm so grateful for all the opportunities you brought to my doorstep. I'd also like to thank my PhD committee of Dr. Mike Schmally, Margie Alexiak, Andrew Baker, and Alex Wilson for their critical insight and diverse expertise in helping me improve every aspect of these projects. I'm really grateful for all of your support and guidance. I'd like to thank Anderson Mayfield as well as the folks at the NMMBA for help completing this experiment in Taiwan, especially Dr. P.Jen Yo, who served as my fellowship sponsor, and especially Crystal McRae. Without any without whom none of this work would have been possible. She's been just an incredible resource and an ally and has saved my butt in so many different ways with these projects. And Ray Tan, who was helpful in the field work. Dr. Stephanie Rosales, Anna Palacio, and Ross Cunning have helped with various aspects of the data analysis and experimental design. And Anna and Andrew were really helpful in giving me access to these Panamanian Pocillopora corals, and Phil Gillette has been caring for them at in the MTLSS aquariums for quite some time. And I want to also acknowledge my funding sources, the Graduate Student Fellowship, the EPSI Fellowship that sent me to Taiwan, and the Rasmus Dave and Roland Fellowship, which supported the bacterial sequencing costs for my most recent experiment. I want to thank also all of the current and past students of the Cnidarian Immunity Lab, who's made this process of helping start the lab and be the first student to proceed through these stages all the more impactful, as I've been able to see how great scientists have followed me and have amazing projects on their own. And I'm so excited to see their defenses in the next year or so. And then I also want to thank the MBE student community, who has always been there for me and has made being a graduate student here at Miami as much fun as being an undergrad was. So this is the Cnidarian Immunity Lab alumni and current virtual class cohort. And then these are some friends from undergrad in the Aquarium Club, which was my core group and helped me get that bachelor's degree. And then the awesome Taiwanese hosts from my summer experience there, without which I would not have been able to survive. I also want to thank my family, especially my brother and sister, for being with me on every part of this, from the first days volunteering at the New Jersey State Aquarium to moving into my freshman dorm and every step in between. You guys are so amazing and have always been so supportive. And then I want to give a very special, unique shout out to the one and only Anne Barkley, who is defending her PhD on African dust transport to the Amazon and tropical Atlantic oceans tomorrow at 11 a.m. So I would encourage you all to tune into that. But Anne is just such a special person and is so dear to me in my heart. And meeting her in graduate school and having our relationship has buoyed me in so many ways. And I'm so grateful to her for her support. And with that, I'm going to open up the floor to any questions, and thank you all for your attention.
Yay!
Woohoo!
Mike, do you want to unshare so we can see everybody?
Okay.
So we can open the floor. Does anybody have any questions for Mike? I have one.
Ask away, I'm happy.
So Mike, how are you reconciling the differences that you saw in the antibiotic experiment only versus the Taiwan antibiotic experiment? Because I feel like what you saw in your experiment that you did at Rasmus was actually a stronger, more pronounced difference than what you saw in the Taiwan experiment. So can you elaborate on that a little bit?
Yeah. So I think that better signature of reduced diversity comes from several lessons I learned from what I did in Taiwan.
Mm-hmm.
And namely, I think it comes down to the use of filter sterile seawater as opposed to 1 micron filtered seawater in the daily media changes that I was able to do in Miami, and having a general understanding of how these antibiotics could shift the coral bacteria community. So I was expecting to see a signature of reduced diversity because I was adding an additional antibiotic with a different mode of action and doing the entire treatment in filter sterile seawater. And so that was the methodological improvement. And I think in the future we should keep that in mind.
Yeah. Do you think that there's going to be a huge difference in the transcriptome versus what you saw in the Taiwan versus? I know you didn't show that, but I'm just curious.
Yeah. I think we're going to see a lot of the same genes activated. What I'm really curious and why I did this experiment was to increase the genotypic diversity. And so I'm really interested to see how the host gene expression partitions according to each genotype, because visually different genotypes responded differently, and they look differently. Like the polyps look like they're almost different characters. And so I think looking for those patterns in the host gene expression and then doing some correlative analyses that link host gene expression to the observed changes in bacteria community will show us across genotypes which genes might be interacting with the associated bacteria. And what it sets up is down the line functional experiments that have more control over which bacteria are present or not present and which host factors are recognizing and mediating those interactions.
Thanks, Mike. Anybody else have any questions?
I do have a question, Mike.
Go ahead, Ben.
Did you look at any of the genes which were driving the difference between your genotypes? So you had like the genotype splitting on the principal components. Have you looked at any of the genes which can be like driving those differences or the processes?
I think that's a great question. I think it's an awesome question. I think it's a little tough to really, and this is why I wanted to do an experiment with triple the number of genotypes. I think it's tough to really draw any meaningful conclusions from comparisons of just the 4 genotypes in the treatment. And so while I was mostly, because there was such a strong sort of additive treatment effect, I spent most of my time analyzing those differences. So I can't say off the top of my head right now which genes are resulting in those genotypic variation. But if memory serves, there were some D-amino acid oxidases that were among the most variable. There were some other peroxidase type genes. So there's these genes that are maybe involved in like epithelial processes that could be what's driving those genotypic variability. But I'd have to look at it again to really know.
Other questions?
Hey, Mike, nice job. I had a question about the results of that final experiment of the antibiotic treatments, and was wondering if you thought that the bacteria that survived were actually resistant to the antibiotics, or if they just were not exposed to the antibiotics because you mentioned that they live deeper in the coral typically. I know it's hard to culture some of those species in the lab, but I guess I was wondering if you tried to culture them on antibiotic plates, if they would survive.
I think that's an awesome question. And I think if I had another 2 years to really work on this experimental system, that would be one of the first orders of business in terms of trying to now go back to these genotypes that we have sort of a baseline diversity from and see which bacteria can we actually coax out of the system and into isolated cultures, because then it would be great to re-inoculate bacteria or a consortium of bacteria to see if we could maybe rescue the bacteria community, post after we stop antibiotics. I think it's possible that some of these bacteria have antibiotic resistance factors, which is why I tried to use antibiotics with different modes of action. So ampicillin blocks cell wall synthesis, streptomycin blocks protein synthesis, and ciprofloxacin blocks DNA replication. So I was expecting to see an even greater reduction in diversity, especially of like the endosymbiosis, because we know that the cultured strains of endosymbiosis are all susceptible to these antibiotics. But when they weren't, when endosymbiosis and some of these other taxa were not eliminated or increased in relative abundance, I had the idea of, well, maybe these are somehow shielded from these external stressors or antibiotics that only make it to, say, the gastrovascular cavity because they're in some sort of internalized niche. So I think to answer that, we'd really need to do like fluorescence in situ hybridization and histology to say, hey, these bacteria are in these aggregates in the gastrodermal cells, and we need to try some other more targeted approach to eliminate them and test their functional roles. So I think, you know, that's where this is heading ultimately. Yeah, thanks for that.
Any other questions? All right, well, let's give Mike a round of applause. Amazing job, Mike. So proud of you. I think we'll take like a 2-minute break, and then the committee, if we're gonna do it on this link, is that?
Mm-hmm.
Okay, if the committee just wants to come back in 2 minutes, get a drink of water, whatever, and we'll go for there. Thank you everybody so much. Congratulations, Mike, wonderful job.
Mm-hmm.
Yay.
Oh boy.